I'm now at the Xradia booth with Kevin Fahey, Chief Material Scientist, and he's talking to us about 3D X-ray microscopy.
Good morning. So as we were speaking just a minute ago, really Xradia's position here in the microscopy community is to really fill in the length scale gap between the traditional CT systems and the electron microscope systems, both of which can give very nice 3D images. But there's this length scale of primary relevance to both the biological and materials community that consists of features in the micron, submicron, and tens of nanometers length scale. So really our focus historically has been to provide system architectures that allow for people to flexibly look at samples with that resolution range. So it turns out that actually the biggest resonance in the microscopy community of late has been the realization that because we can do that imaging non-destructively, you now end up being able to use the techniques such that you can do all of your imaging work. You can do four-dimensional and time-dependent studies, or even in situ studies inside of a system, all non-destructively prior to then sending the sample into the electron microscope. So if we look here, for example, these are two of our laboratory system architectures, right? The Versa architecture, which you see here, is one that we use in a great deal for these sorts of in situ experiments. This is actually a rat jaw in a water solution under tension and compression for a cyclic fatigue study. And so being able to do these time-based studies and look at how the microstructure evolves ends up being critical, particularly, as I said, before you go into something that's destructive but perhaps can give you a richer quantitative data set like a FibSim or dual beam. In between this and the electron microscope lab fits the ultra style. This is a synchrotron nanofocus style architecture. So here you're actually looking at monochromatic X-rays with Fresnel zone plate imaging objectives, and actually the ability to focus in and get very high magnification in the 50 nanometer or 100 nanometer resolution range. So really all of these together are now coupling into the ability to really start with cell phones and coffee mugs and full embryos and work your way all the way down into the high resolution electron microscope range.